both microorganism, microbiota and microbiome, right? So we already walked through about the microorganism, microbiota. When we come about the microbiome, it is the study of the microbiota and their genetic, right? So we focus on their isolation. And when you go for the genetic environment, that says microbiome. And the metagenomics is a technique, okay, where you use to sequence those genomic DNA or RNA. Okay, whatever the DNA you extract from, wherever you can collect the samples from here or anywhere from the world or other universe, right? They sample, extract DNA, and analysis is called metagenomics. Okay, these are the terms. So now coming to that point, we call the great plate count anomaly. What I said previously, when you take the samples from any environment, that from space, International Space Station, right? You have been watched so many movies where they taken like what movie? The James Ravi. Tick tick tick, right? Tick tick tick. They went to the International Space Station, right? To refuel the vehicle. You don't know that? Okay. And that is from ourselves. Not only from ours, honeybee, what else? Anything. If you want to check their gut, that is also be part of it. And metasub is a metagenomics of subway. Okay, they started their analysis from New York subway. Now we are also doing in India, Russia. So we are the one of the part of this metasub. Wherever there is a metro system in India and metro urban city, so we collect the samples from there and analyze the metagenomics of an organism. And Illumina is one of the platforms they use for next gen sequencing, right? To understand those diversity of microbes. And King Abu Dhabi University of Science and Technology, Coast University, they focus on coral metagenomics. Okay, coral reef. If you deep dive into the Red Sea or any sea, you can see the coral, right? How the microbiome will help them to grow the coral. Okay. So now we took the samples, and some people are doing traditional way of microbiology. So they are plating on any plate, and someone are doing microscopic stuff. Okay, when it comes, you can see colony gives only 5 colonies, but you see 1, 2, 3, 4, 5, 6, 7, 8. 8 different cells are there. Where the 3 went? What happened to the 3? Where we are missing? Where we are missing the dots? We are unable to connect the dots, right? We need to connect all the dots. How we can connect those dots, right? So why few of them are able to cultivable? There are only 1% of total microorganism can be cultivable in a laboratory condition. Rest of 99% of microorganism cannot be cultivated. Maybe you have to modify multiple things. Means the media have to be modified, the temperature control, the pH control, the environment. See, from the very sample, you cannot be able to give the same environment, grow the microorganism in the laboratory condition, right? So then how you are going to understand those microorganisms? The way you can do cell counter, short them, and understand the genetic material. That will help you. So there comes the cultured independent techniques, right? Until you see, we know all this thing, right? We will talk Robert Koch, right? Those are traditional way of microbiology. And when they initially start the first DNA sequencing, initially they focus on the 16S ribosome RNA, right? So they sequence the 16S and they understand the diversity. Previously we start with the morphology and went to the 16S. Now they use the same 16S, deep sequence them, okay? Deep sequence with the help of next generation sequencing. We will go in the next slide in depth of this next generation sequencing, okay? So this is again a map from our gut, okay? So whenever you do some research on organism, you have to give the diversity, where it comes from, how it is diverse from the environment. Okay? We cannot simply say this is the organism that isolated, this is the bacillus organism. Right? Nowadays they made an organized structure if you want to claim that is organism from this organism. This is my organism. No, you cannot do within a morphological trait. You have to deep dive into the molecular biology and the whole genome sequencing to understand this is my bacterial species. So what we did, we isolated. I will go to the story later and I will ask you a question. You will learn about that one. So we gave one organism that was isolated from International Space Station, that was unique, novel, and we gave, my mentor honored Dr. Abdul Kalam and gave the name on behalf of Dr. Abdul Kalam. Okay? So you can find that organism. When I come to that one, you can say that organism. Okay? So this is how it is diverse from our gut, and how it will be diverse in another environment. Right? So here is some other snapshot that will help you to understand. Okay? So think about the red bacteria that is closely related to our chromosome, that can integrate in our chromosome, so that we call as a post-symbiont genes. Okay, some blue that can co-evolve with the symbiont gene, that will co-evolve with us, some organism. Some organisms do not affect our phenotype. Some organism will affect our phenotype. Means someone will feel that I want to have a chocolate. That the mood swing can be done by your gut microbiota. This is the research is nowadays going on. When is my mood swinging? Do I want to eat chocolate now or not? Your gut microbiota is deciding. Is it true or false? Means people are asking question whether my gut microbiota making me to think to have this chocolate now. Right? So that was into our system. Right? So that's my background of what I want to give you from the metagenic perspective. So now, when we want to do something in the International Space Station, we want to understand how many organisms are viable. Means when you stick the plate, you see only 10 colonies are viable. Rest of the 10 colonies are viable or not. We ask this question, whether the other 10 are viable or not. So we use some technique called Propidium monoazide treatment, where this dye will go and bind to the dead cell, non-intact, compromised cell, and binds to the DNA. Okay? Now, when you PCR them, you see PCR, whichever the DNA has the PMA, it won't give any amplification. Okay? So now you see it is a compromised DNA, then the PMA will get inside and there won't be any amplification. Means there will be an amplification when they are having an intact DNA. So the DNA was isolated from the viable cells. If you have a sample, an A sample and a B sample, want to see how many of viable cells and non-viable cells. So you will treat with the Propidium iodide and see how much is viable, how much is non-viable. Likewise, rapidly you can assay with the ATP. So ATP, you know the biomolecule, which is energy-produced biomolecule, and every organism will produce this ATP. So when you identify ATP from intracellular, then it is viable cells, intact viable cells that can be able to give successive generation. If the ATP is from outside, that is not viable cells. Okay. So let's come up with the evolution of sequencing here. So first, from this angular sequencing, this is the first generation where they identify only the 500 to 1000 base pair fragments. Okay, they use these techniques. We are not going to deal with the chemistry of the sequencer. Just giving some information to you. First generation is for the Sanger sequencing. Second generation is Illumina. We call it next gen sequencing, the high throughput sequencing, where it will help to give only this much fragment, but it will go to the deeper. Means here it is 500 to 1000 base pair. You have to give a specific amplification. There also you will give, but it will generate lot of data compared to the Sanger sequencing. But coming to the third generation, PacBio and Oxford, these are short weeks, means 50 to 500 days, right?